Okay, I think we're going live. So it is Sunday, um, July 15th, and I had a couple of questions about this week's information, so week 8, and then I thought I would go ahead and do maybe a little bit of lecturing on week 9 to kind of tie things together. So let me start. I've already pulled up I don't know where I went. I guess it's still recording me. Where'd I go? There I am. Okay, let's see where I can put me. I'm going to move me across. Might get a little bit dizzy there. All right, so let me pull up this week's materials. So chapter 14 and 15. Here we go. I'm going to go back and let's see if we can do the slideshow here. Maybe that'll make it easy. There we go. All right. So a couple of you, um, this unit, so we get out of mitosis and meiosis, okay? So when we talk about mitosis and meiosis and the labs for those, we're talking about simply for mitosis, we're talking about making more, right? So what is the purpose of mitosis? It is just to, if you have a cell, it is to start out with 1 and make 2, you have 2, you make 4, et cetera, et cetera, et cetera, right? The idea is that everything is identical. And then with meiosis, we have the gametes, right? So those are the sex cells. Um, and the point of those, so with meiosis 1, the point of that is to have crossing over, right? So we want some diversity. And also, we want to make sure that the number of chromosomes, like for example, in a human, goes from 46 to 23. So we have, for example, we're diploid and you go to haploid. So, and then the second part of meiosis, meiosis 2, pretty much looks like mitosis. The point of it is to make more, okay? So, and a few comments on those when I was looking at the labs about mitosis and meiosis. So there's a couple of, mostly mitosis and meiosis, there's a lot of definitions. So you have chromosomes, tetrads, the different phases. So prophase, metaphase, anaphase, and telophase of mitosis. And then you go into meiosis and you have meiosis 1 and you have prophase, metaphase, anaphase, and telophase all with a 1. And then you have meiosis 2 and you have prophase, metaphase, anaphase, and telophase. Okay. And so I saw a little bit of confusion with maybe meiosis 1 and meiosis 2. Keep in mind that for meiosis, again, the whole point of meiosis 1 is to have the information and to diversify it. Okay, so we're not trying to create, like we are with mitosis, we're not trying to create cells that are exactly the same, right? So you have crossing over that occurs in prophase. Okay, and I'm not asking you to go into a huge amount of detail about crossing over. It's actually a very complex process, um, but we're trying to kind of keep it, at the 10,000, 20,000 foot level. And metaphase means middle, so everything lines up in the middle. Anaphase, ana means separate or without, so it separates the chromosomes. And then in telophase or telophase, you have half, in the case of meiosis 1, you have half of what you started out with, plus they don't look like the original because crossing over has occurred back in prophase. And then it goes right into meiosis 2. Okay, there's no stopping, it just keeps on going. Okay, and then meiosis 2 looks very similar to mitosis, except for the fact that you now have these diversified chromosomes that are being pulled apart. Okay, so that at the end you end up with more. So the whole point of meiosis 2 is just to end up with more cells, right? If you start out with 2, then you're going to end up with 4, okay, when it's all said and done. So I just want to make sure that that's clear for everybody, because otherwise, when you go from mitosis and meiosis to genetics, um, you might be a little bit lost if you don't have the basics down pat for mitosis. And you can go into the information with mitosis and meiosis, And there's lots of good things online and in your our class that have to do with, you know, what mitosis is, what mitosis isn't, what meiosis is, and what meiosis isn't. So you want to make sure that those are very clear um, in your head, especially before you go into what we're going to talk about now, which is genetics. So this is my favorite, my favorite part here. And this is a picture in front of you of Mendel, and Mendel was an Austrian monk working on pea plants. And he is known as the father of Mendelian genetics. Okay, so let's kind of go through Mendel. Whenever you start out talking about genetics, okay, you really start out with Mendel. So it's kind of like the beginning, okay? So we're going to base everything in terms of hypothesis testing, right, on what Mendel saw, his observations. And Mendel was not some great scientist or anybody like that. Like I said, he was an Austrian monk, and if you know anything about monks, they have a lot of time on their hands. So he used something that was readily available. It's not that he knew a lot about it or he knew that pea plants were the plant to use, but he actually kind of got lucky, okay, in one sense, and he did something that a lot of other people didn't do, and that is that his experiments were very, very carefully planned, and he wrote everything down because we have a saying that goes, if it's not documented, it didn't happen. Okay, so this is just a slide basically showing you what he did. So you have a purple flower. You know, there's many different traits for the pea plant, as you know, but he took the stamens from the purple flower and he transferred some pollen, you know, with the white and the purple, and you had the pea generation, okay, which is not P as in PEA, but parental generation right here. Okay, so parental, it means the beginning. Okay, it doesn't necessarily mean your parents, it means the beginning. So he crossed a purple flower with a white flower, and this is what it's showing. Okay, you have removed the stamens from the purple flower, transferred the sperm-bearing pollen from the stamens of the white flower to an egg of the purple, or vice versa. In his case, you know it doesn't matter. And you got your peas. So with peas, these little round peas are like the offspring, okay? And then you take these peas, these pea right here, these individuals, and you plant those. Those are the seeds, okay? And so all of his F1, everybody in the next generation, so mom and dad are purple and white, let's say, purple and white flowers. Everybody in the F1 generation or the next generation is all purple. Okay, and this is what he saw. All right, so he then took all of the purple plants and he crossed them to each other. And in the plant world, we call this a self. It means you take a plant and you cross it in the same generation to itself. Okay, you don't hear this term with people or mammals, things like that. You know, we're not really crossing ourselves. But in plants, which makes it nice, you can do that. So if any of you are gardeners or you like to plant, which I did see some um, good discussion boards about flowers, then you should be able to relate to this. And then when you cross those two, okay, he ended up with this F2 generation. So you can see that you just keep going. And what he saw was he saw this 3-to-1 ratio. So this is not just a picture. This is actually what he saw. And there was basically 75% were purple and 25% were the white flowers. So they're like, you know, that's interesting. He didn't know the reason why. He called it kind of this either or, you know, it's either purple or it's white, right? So in the parent generation, again, you have true breeding parents. That's the other thing that, you know, we would learn that he saw, in that this wasn't a heterozygote and this wasn't a heterozygote. These were homozygotes. Okay, so homozygote means same. So you can imagine that this is, we'll say, purple, purple, and white, white, and then everybody in the F1 generation was purple. And then in the F2, 75% was purple, and a quarter of them were white flowers. Okay. So And he didn't know, this is the law of segregation. Like, he didn't know, you have to keep in mind that what we know today about genetics, and we have all this molecular stuff that we can do, and they didn't know that back then. So in 1909, Mendel didn't know any of this. He just wrote down what he saw. Okay, so we have Mendel's model here, and these are his actual numbers that he saw for the F2 generation. Okay, so you can see that it's not exact, but you do see that there is that 3-to-1 generation. Okay, so you see that he looked at flower color, flower position, you have the axial and the terminal buds, you have seed color, seed shape, pod color, and stem length. So they're all pretty self-explanatory. So peas were a good model organism, and again, not even in the sense that he knew, that, oh, I'm going to use a model organism. He didn't know that, but as you can see, he was able to say, okay, and he coined the terms dominant and recessive. So dominant simply meaning something that you see a lot, and recessive meaning something, in his case, that if you remember back to the purple and white example, in the first generation, so in the F1, he saw all purple, right? So that became the dominant, and then in the F2, the white trait came back, which meant that that was recessive. So those were the terms that he coined, okay? All right. So then we kind of move on and we talk about something we term allele, okay? So a genetic allele. An allele is, you can think of a allele, it's just an alternative name for a gene. So you could call, so we have purple and white again. So you have the purple gene and the white gene, but in genetics we refer to them as alleles. Okay, so you have the purple allele and the white allele. Okay, and you know that chromosomes are homologous, so this would be a pair of homologous chromosomes. And if you have purple on one and white on the other, then you're looking at a heterozygote, because hetero means different. Okay, if they were both purple, they would be a homozygote, dominant, right? Because purple is dominant to white. And if they're both white, then they would be homozygous recessive. So we start getting into in genetics. I would say 80% of genetics is probably the nomenclature. So don't let the nomenclature confuse you. You want to make sure that you have that straight in your head, so that when you're kind of get into the more difficult stuff, it doesn't throw you. Okay, so moving on, and you already know this, that there are 2 alleles per organism, right? So they can be, in this case, the 2 alleles at a locus on a chromosome may be homozygous, right? They might be the same, or they might be different. So this is not telling you anything different than I didn't already say. Okay. Then we get into dominant and recessive. So dominant, you know, that is usually going to determine. So again, this is in Mendelian genetics only. Okay, and I can't stress that enough, because when you get into the more complicated genetics, the rules change. But we use Mendel's laws, right? Law of segregation, law of independent assortment. We use the Mendelian rules as our null hypotheses in genetics, because again, like you have to have something. Null means it's not changing. So if Mendelian genetics is kind of the base and everything else kind of sprouts off of it, then it makes sense that the Mendelian, that 3-to-1 ratio that we just talked about, that would become, if you have one trait, so like purple and white is flower color, that's 1. Okay, when you add 2, let's say you have flower color and pod shape, that's 2 traits, so that's dihybrid. So monohybrid is 1, dihybrid is 2, and that generation where we were crossing the heterozygotes, that's where that name comes from. Okay, so we'll talk more about that. All right, so the first, we get into the law of segregation. So the law of segregation you know that you have two alleles for a heritable character. Okay, that just simply means here's a homologous pair of chromosomes in a diploid parent cell. Okay, so this, they're going to have replication. This should look very familiar to you. You have meiosis 1, right? It splits. And then in meiosis 2, it just makes more. Okay, so we haven't gotten into crossing over and all that. This is just simply states law of segregation. Okay. All right. Now, let's talk about a Punnett square. So some of you have asked me about Punnett squares. So Punnett squares, I like Punnett squares because I'm very visual, and this gives you a very visual representation of what's happening. I think this is the only way I made it through genetics, to be honest with you, at least in the beginning. So let's just go right back to what Mendel did, right? So we already know that he crossed purple flowers with white flowers. We know that purple is dominant to white. We know that he had true breeding plants, which means that double P right here, right? We've got homologous chromosomes, right? And then we've got little p over here, right? Okay. They cross, you get your F1 generation, everybody's purple. So purple, then we introduce kind of two more words, all right? So we've got phenotype, which is what you physically see. So the phenotype of the F1 generation is all purple. But the genotype of every single one of those flowers in the F1 generation is big P, little p, which means they're heterozygote, okay? So they have half of purple dominant and half of the white little. So in other words, that's what they can give in their gametes. Okay, so keep in mind that we're talking about what an egg and a sperm could give. So this would be called, in this case, this is a monohybrid cross right here. Okay, you have one trait, one purple flower, okay, one flower color, okay, and they cross them to each other. That's called a self. And you get, so here would be the male, right? You have, he can give a big P or a little p, and then you have the female, she can give a big P or a little p. And that's all coming from this, okay? And when you cross them, you simply are multiplying. So if I take big P and multiply it with big P, I get big P, right? Or homozygous dominant purple, okay? And then you take the same thing, so I'm going to bring my P over, and I'm going to bring my little p down, and I'm going to get purple in terms of a phenotype, but my genotype is going to be big P, little p. Okay, it's a heterozygote, right? I'm going to do it again. I'm going to bring in the little p. I'm going to bring down my big P. I'm going to get the exact same thing. So 50% of my offspring are going to be big P, little p. A quarter of them are going to be homozygous dominant, and a quarter of them are going to be homozygous recessive. Now that's genotype. That's this. This is a genotype. In terms of phenotype, 75% or 3 are purple, and 1 or 3 to 1, 3/4, 1/4. All this is a ratio, and there's many different ways to have a ratio. I mean, I could say that this is 75%. I could say it's 0.75. I can say 3/4. I can say 3 to 1. So keep in mind that these are just mathematical terms. They're not saying anything different. It just depends on really what the person wants to put it in. Whether they want to do 3 to 1, 3/4 to 1/4, or in percentages. Okay. All right, so here's another way of looking at it. It breaks it down very nicely. So if you're having trouble with phenotype and genotype, and the F2 generation of what Mendel saw for a monohybrid cross, come back to this slide. This is a good one. Okay, it kind of breaks it down really nicely. All right, so the monohybrid, so everything we've just gone over is monohybrid cross, okay? So that was one type of cross that he did, okay? He did quite a few crosses. Another type of cross that he did is called the test cross. Now you may be asking yourself, well, why do you do a test cross? Well, you do a test cross when you don't know, so you always know what the recessive type is because it takes 2. So when I think recessive, I think it takes 2, right? You have to have 2 little p's to be white. But to be purple, if you remember correctly, you could be homozygous dominant or you could be heterozygous dominant. So even though you have a dominant phenotype, okay, but let's say you want to know what that parent was and we didn't know whether it was homozygous dominant or heterozygous, because when you see purple, right, you don't know. You just know that it's one of these two. So you want to find out, okay? So how you find out is you take the recessive that you know. It's something that's known, okay? So it's at the top here. And you cross it to that purple flower, okay? And if everybody is purple, okay, then the parent must be homozygous dominant, okay? And that is another way of saying true breeding, okay? And that's what he did, and that's how we figured out that he was using for that original parent generation that purple flower was big P, little big P, big P, okay? Now, let's say that he had done this and for that parent generation and he crossed with the purple, okay? And he had gotten a 50-50, okay? Well, if he had seen 50-50, then mom would have been a heterozygote, okay? So a telltale sign, all right, so the question is, well, how do I know by doing a test cross that mom is either, or dad, in this case it doesn't matter, but how do I know that she's homozygous dominant or she's heterozygous? Well, how you know is this right here. Because if she's heterozygous, you're going to see a 50-50 or a 1-to-1 ratio of the offspring, purple to white. If she's homozygous dominant, right, everybody is going to be dominant. Okay, does that make sense? Okay, and if and again, this is a in this particular case, this is talking about, you can look at this, well, how would we know, or how did Mendel know that his parent flower that he started out with that was purple was true breeding? Okay, all right. So moving on to the law of independent assortment. So Mendel is credited with two laws, law of segregation and the law of independent assortment. And so we kind of harp on those in genetics because, again, as you know from this class, in biology, there aren't many laws. Okay, so physics has the laws, biology doesn't. Biology has lots of theories. So they have a lot of evidence, but not everything is, there's a lot of hypotheses and a lot of evidence and a lot of support, but there's a big difference between something that's a law and something that's a theory. So now we have law of independent assortment. So we'll just use two other characters, okay, and this gets into his dihybrid. So to recap, the law of segregation was determined by a monohybrid cross, okay. The law of independent assortment is determined by a dihybrid cross. And then if you don't know one of the parents or you're trying to figure out, let's say you're in a field and you want to know, well, what's the genotype of one of my flowers? We're crossing a white to a purple, and you know that the purple is dominant, and you want to know what is it big, homozygous or heterozygous, then you can do a test cross and find out. Okay, so here we have another P generation, right? We're going right back to the beginning. And in this case, we have two traits. Okay, so we have color, right? This is yellow and green, indicated by, we'll say yellow is big Y and green is little y. And then the other trait we have is round, which is a big R, and we have wrinkled, which is a little r. Now, again, we know, he did the work and he did test crosses and cross these many, many, many times. So when he first crossed these together, okay, the gametes could be this either dominant, right, and recessive. You put them together and you get big Y, big R, little r. So this is a dihybrid. Okay, monohybrid is 1, dihybrid is 2. Okay, these are true breeding. That's true breeding. This is true breeding. True breeding means it can only give 1. Like, so this can only give a big Y and this can only give a big R. And if I had 5 more traits on this, then they would give, as big M, then it would only be able to give an M, okay? And true breeding here, right, means it only gives 1. So now everybody in the F1 was dominant, so everybody here looks round and yellow. But again, so then the question becomes, well, are they independently assorting? If you have 2 genes, right, are they independently assorting or are they dependently assorting? So dependent would mean they would go together, okay? So kind of like linkage or something like that, right? If something's linked, it's going to go together. So what he saw was that they were independently assorting, so there was a 25% chance that a sperm would be dominant, completely dominant. Here we have dominant plus recessive, then we have the other way around, and then a quarter of a chance of being completely recessive. And this is where, when you put all these in, okay, so dad gives the same thing as mom in this case, okay, because they're plants. There's no gender bias here, okay? And this is not the case, again, with a lot of other things, but in Mendel's case, we'll take it. And so when you do the math for all of this, okay, there are 16 squares, okay? So that's where this is coming from. 9 of these 16 squares will be completely dominant, okay? So they're going to be yellow and round, okay? 3/16 of them are going to be green and round, okay? The other 3/16 is going to be wrinkled and yellow, okay? And these are interchangeable. It's just a 9, 3 to 3 to 1. And then 1 out of the 16, that should be 1 out of 16, is wrinkled and green because those are recessive. So it's a 9, 3 to 3 to 1 ratio. And if you add those up, 12, 13, 14, 15, 16, you get 16 out of 16, which is 1. Okay. Let me put this on pause for one. Okay. So some of this I've already gone into. This is just more of an explanation of how this works. So if you're having trouble trying to figure out probability, there's a little bit of math involved in genetics. Doesn't get too complicated, okay? But it just explains to you with coins, because we're all familiar with coins, hopefully. And, it's kind of like flipping a coin, right? You have a half chance of getting a head and you have a half chance of getting a tail, okay? So, and then with the other side, right, mom, dad, you have half a chance of getting a head and half a chance of getting a tail. Okay, so this is what, you're putting them together. So the segregation and independent assortment. So you've got the fact that they independently assort into the offspring, and then you've got the chance that they segregate into, like this sperm, for example, has a half a chance of being, in this case, a head, and a half a chance of being a tail. Okay, so this is the segregation part. And then they independently assort into the offspring, right? So if you were to say, for example, well, what is the probability that my kid, my offspring, my pea plant, whatever, will be homozygous dominant, then the answer is a quarter or 25% or 1/4, depending on how you want to look at it. But if you're talking about one trait, it's all out of 4. So because you can't have more than one, right, like 100%, when you talk about probability. So if you're coming up with answers that are over 1, it kind of tells you that something's not right in the math part. All right, so it gets a little more complicated when you have multiple traits. Okay, so like this one, for example, is dealing with three traits. So you have the P's, the Y's, and the R's. But you see that you can look at, they're still independently assorting if the trait is Mendelian, or if all of the traits are Mendelian. They're still independently assorting. So it's like, what's the chance of at least 2 recessive traits? You're figuring out how to come up with it. Okay, so it's the product of multiplication with just multiple characters going across. All right. So let's see. So we get into, so here's a pea plant. It's really a simple cross. You've got either a monohybrid, dihybrid, or a test cross, right? Well, a lot of things, I would say most things, are not as simple as Mendelian genetics. So again, Mendelian genetics is the basics. But the laws of segregation and independent assortment apply to everything. Okay, it either independently assorts, right, or there's something else about it that's different. Maybe it's linked, or something is different. Okay, but that's how these became laws. I mean, you can apply them to really, really complex ideas. Okay, so let's take Mendelian. Okay, so we have our basis, all right? We're kind of building, it's like building a house. Okay, so our foundation is Mendelian genetics, all right? And then we're going to say, well, what happens? We're going to kind of build up from there. Well, what happens when it doesn't follow Mendelian pattern? Freak out and don't talk about it. No, you have to have a way to explain it. So, what are some reasons that they might not be Mendelian? Well, maybe the alleles are not completely separated, right? They might be linked. Maybe when a gene has more than one phenotype associated with it. You get into like when purple and white make lavender, or purple and white become spotted or something like that versus just purple and white. So those are some examples. So for example, these are always kind of interesting, but again, they're more complex than what Mendel saw. He got lucky. He really did. I know it kind of sounds cheesy, but if he had been working with, let's say, a different organism, all right, and it hadn't been peas, he may have seen something like this, right? So here we have red crossed with white. Okay, so those are still true breeding, right? They've only given one. And suddenly, in that F1, they're all pink. Whoa, that would have probably blown his mind if he had seen that. So it's like, oh, whoa, wait a second here. So we get down and we say, well, what's happening? We say, well, they're still giving. So in terms of genotype, right, still bring down the R and still bring down the W, right? So here's female and male. But when you have the homozygous condition, you see the dominant is definitely red, right? I mean, that's right. And then the recessive is obviously white. But when you have that heterozygote condition, you have a third phenotype, which is pink. Okay. All right. So I'm not sure what this dominant allele. It basically, the relationship between dominance and phenotype. So dominance, um, something can be dominant as long as something is dominant to something recessive. It has to do with how you see it. All right. But when you see something that might be co-dominant, okay, that means that both colors are going to come out. If something is incompletely dominant, then you don't necessarily see one or the other, right? Okay. So it just depends on how far apart the genes are, how they are, are they linked? Are they not linked? Distances. It's just more complex than, simply, well, this is tall, or this is purple and this is white. Like I said, Mendel got lucky. So, for example, this is very interesting that dominant alleles, so you don't think of dominance as being necessarily the most common. It just means in terms of the genes that it's dominant to something else, right? But there's a lot of something else's, okay? There's something else that could be dominant to it. So for example, one baby out of 400 in the United States is born with extra fingers or toes, right? This is the polydactyl. Well, interestingly enough, it's the more fingers and toes, it's the polydactyl trait that's actually dominant to the allele for the more common 5 fingers and 5 toes on each hand or foot. Okay, so in this case, right, the recessive allele is actually more prevalent than the dominant allele in the human population. Okay. All right, so we get into sometimes you have multiple alleles. All right, so what is a multiple allele? Well, blood type is probably the easiest, most concrete example of multiple alleles. And so you say, well, I don't understand that. How can that be? Well, because as you can see, right, you can be blood type A, you can be blood type B, you can be blood type AB, right? Or you can be O, which is actually recessive. So that's what this is showing you. So when you are A, so if you have the blood type A, you can either be homozygous A or you can be heterozygous A, right? Same with B, and then AB is codominant, right? Because you're going to see both A and B here. And then you have recessive, okay? And these are multiple alleles because obviously there are A, B, AB, and O, okay? All right. Another effect, another phenotypic effect, if you will, is called pleiotropy. And pleiotropy, when you see something that's pleiotropic, these are going to be the genes that have very different, almost like an assortment, if you will, of phenotypic effects. Okay, so for example, cystic fibrosis is a good one. There are so many different mutations in cystic fibrosis. It is not a one or the other. Um, but the genes that confer cystic fibrosis, okay, not everyone who has cystic fibrosis has the same symptoms. That's what it's saying. Okay, they vary, and with sickle cell disease, okay, so something like sickle cell anemia, that not everyone who has sickle cell has the exact same. It's not like you can line everybody up and be like, oh yeah, everybody has the exact same phenotype. Everybody who has sickle cell doesn't have the same number of sickle cell shaped red blood cells. Okay, so that's what that means. It means multiple phenotypic effect. Okay, you can think. Let's see, another example that's not kind of like these would be where you can see you might have a gene for color, and it might be like dark purple, medium purple, light purple kind of thing. I mean, they're still purple, but they're not always the same shade. Okay, so then we kind of get into, so now we keep going, we got to get into, we're extending Mendelian genetics, and we're talking about 2 or more. So this is pretty good. Like you got the 2 parents up here, you've got your little mice, and this technically would be considered a dihybrid. Okay, so that, and when I say dihybrid, I'm just saying that it's 2 genes, right? I don't even know what they are. I mean, I really don't need to know what they are. You know, you got a B allele and you got a C allele over here. And so you have your independent assortment, right? Okay, so nothing new there. And then you have your independent assortment, and you have 16, right? So this is a dihybrid cross. You could set it up and you could say, without looking at any of this, you could say, well, what do you expect? Okay, what is your hypothesis for this? Well, your hypothesis is 9-3 to 3 to 1, right? Because that's a dihybrid cross, okay? But instead, when you actually cross these and you see what the phenotypic ratios are, you see that they are not 9-3 to 3 to 1. Okay, you've got the 9 for the dominant, and you've got the 3 over here, but then there's a 3/16 that's missing, and then you have a lot more recessive than you should. Okay. So then we get into something, so we're kind of taking it, and then we get into something called polygenic inheritance. So skin color is probably the best example of polygenic inheritance. There are many, 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 many genes involved in skin color. Okay. And so as you can see here, you're going to put lots of genes together. I mean, in reality, it probably goes out farther than this, A, B, C, D, E, F, G. And you can see the differences between, like, this would be like really, really, really pale, 1/64 over here all the way. And this is literally like color. And on the other side, you have, kind of like your two ends of the spectrum, right? Like a normal distribution, if you will. Okay. And so this is an additive effect, right? Like where you have two or more genes, and where this phenotype that you see, there's a lot more that going into that phenotype than just this or that. Because remember, the only thing that's this or that is Mendelian. So you got the environmental factors. I mean, there's just so many different things that affect why something would not follow a Mendelian pattern. Okay, again, it is all about null hypotheses versus something else. Okay, we're not proving anything. Hypotheses don't prove anything. They're either supported or they're not supported. Okay, so as a geneticist, let's say that I'm looking at a gene and I want to know it's a new gene and I don't know anything about it. Well, the first thing I'm going to do is I'm going to look at, I'm going to do some crosses. I'm going to look at all the little offspring, and I'm going to say, well, is this a Mendelian trait? Okay, well, am I doing a monohybrid? I have to go and I follow the things that Mendel did. So I make a cross of, maybe I do a test cross because I don't know whether, mom or dad is homozygous or heterozygous, right? So that's the first thing. I got to figure out, well, who's dominant to who? Who's dominant and who's recessive? Okay, I'm going to do a test cross. Okay, when I do my monohybrid cross, okay, to get my F2 generation, does it follow a Mendelian pattern? Do I see 3 to 1? If I do see 3 to 1, fine. I know it's a Mendelian allele, it behaves Mendelian. If it's not and it's something else, then what is it? What does it do? Is it codominant? Is it incomplete dominance? Is it multiple alleles? Is it pleiotropic? Is it maybe there's multiple genes involved in whatever I'm looking at? And then let's discuss environment. You cannot have an individual without taking into consideration why you see what you do, without taking in consideration environment. There's environment and there's genetics. So there's the alleles, there's what something is made of, and then there's the environment that that organism is in. And you cannot separate the two from each other, okay? I mean, you can in a controlled environment, but in terms of, natural environment, when they're outside, you can say, well, maybe this one, this purple hydrangea, let's say I have a purple and pink hydrangea. Well, that's influenced by the acid that's in the soil. So you can make different flower colors based on the acidity of the soil. So this environment, when I say environment, it doesn't necessarily mean bad. It just means that there's a change, right? So you might have a genotype and the genotype might be big B, little b, let's say, blue dominant, okay? But I can take that exact same genotype and I can put it in different soil pHs, okay? And I'm going to get 6 different phenotypes, ranging from, purple, purple, all the way to pink, pink, let's say. Okay, so that's what you have to keep in mind when you look at genetics. All right, so here are some inheritance patterns. These are both actually Mendelian. One of them is the widow's peak and the other one is the attached earlobe. So dominant is the widow's peak, and you can see here this is a widow's peak. And dominant here is the free earlobe or unattached earlobe. And so you can figure out, right, by, you can go back. First generation could be your great, 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 great grandparents. It just depends on what you have, right? What information you have. And you draw these out to figure out. And by doing so, you can figure out, well, if this person and this person get together, what will their kids look like? If they're a girl, boy, we get into sex-linked traits. So you have male, female. You'll notice that the names in genetics, you also saw that in the Punnett square, that the left side has the female and at the top is the male. Genetics happens to be a field that has a lot of very feminine names associated with it. You notice in mitosis and meiosis that we talked about sister chromatids and tetrads and sisters and things like that. They have a feminine underlying, like, kind of nomenclature, feminine nomenclature to them. So that's not something that you're, just seeing. But all right, so recessively inherited disorders. I think that this is, if you have something recessive, like for example, cystic fibrosis, you just have to remember that in order to be afflicted with something, that it takes two. Okay, if it's something that's a recessively inherited disorder, right? It takes two. Okay, so if you're a carrier, there's lots of, you have a very high chance of being a cystic fibrosis carrier, but they don't have the disorder. Okay, they don't have it. All right, they can pass it on. If you have two, there's a 1/4 chance, but they don't necessarily have it. And so cystic fibrosis is just really, it's a very horrible condition, and it's, some of this data I think is kind of, I don't know, outdated, but there's more than this. And this gives you, as far as like the symptoms go, this gives you a good idea of what it is, but this is in no way, shape, or form constitutive at all. Okay, this is not everything with cystic fibrosis. Sickle cell disease or sickle cell disorder, again, you're going to have the 1 out of 400, but to have sickle cell, you have to have, this is recessive. So when the sickle cell allele gets into the population, or the cystic fibrosis gets into the population geneticists will follow it because they want to know, are there more people getting it? Is the birth rate of people higher or is it lower, et cetera, et cetera. This is something we don't go into a lot, but you've probably, it's called consanguinity, which is a kind of a mouthful, but this would be like a close mating of relatives. And, people kind of roll their eyes at it and stuff, and maybe it's not as socially acceptable, obviously, today. But back in history, there are several noble families that consanguinity was the way that they went. And so because of that, and they were trying to keep, the idea was that they were keeping the gene pool pure. That if they don't go outside the family, then they don't introduce any unpure genes into the lineages. But what ended up happening with some of these is that they ended up, a lot of those genetic disorders that are recessive, right, they stayed in the families. And one of the biggest ones was hemophilia. And I mean, it just wiped out entire lineages of people because of consanguinity. All right, dominantly inherited and lethally recessive. I mean, there's all kinds of stuff. And you get into genetic testing and genetic counseling. And you have, so the old way of doing it really was the amniocentesis. And this is this big giant needle, and they would take out the fluid from the 14th to 16th week and go through and test it. And this is very risky. This right here is very risky. Now, they can do something a little bit different that's called chorionic villus sampling, or CVS, and it's much earlier, and it gives you the same result, right? But it's the big thing with this is that it's a lot earlier. I mean, they're both kind of risky. All right. So at the end of this, I'll let you do these questions, but at the end of this, there's some questions to ask. Okay. So let me pull up chapter, this is the one we just did, yes, and you can keep going through that. So that's why I was telling you that what am I trying to say? If you pull these slides up, you can really, and you can practice these, and it gives you the answer as you're going through. And so if you're having trouble, you can ask me certain, hey, like slide number 39 or slide number whatever, and I can help you a little bit better, because there's a lot of concepts that are kind of packed in here. Okay, so what I just went over was this past week, so the week that we're in, and 16 and 17 are this weeks that are coming up. So let me go through this a little bit. Hopefully this doesn't get too long for you guys. Okay. Let me get this bigger here. All right. So I'm going to kind of skip through a little bit. All right. So when we get into 16 and 17, so we're going to kind of switch gears. So we kind of get out of pure genetics, okay, and we're kind of going to go into the molecular side of things, okay, and that can be a little bit confusing, but remember, we're just kind of going back and talking about DNA, so that's really all this is about, all right. What's interesting is that it took us until the 1950s, well, it took us until the 1940s to know that indeed DNA was the genetic material, but it took us until the 50s to actually figure out what DNA, or give a model in terms of what DNA actually looked like. Which I find that just funny, because Mendel's work was done in the early 1900s, so this wouldn't come along until about 50 years later. And here is Watson and Crick. And again, like some of the timeline in your class on Blackboard, there's a timeline of DNA being the genetic material. And most people thought that it was proteins that was the genetic material, which makes sense, right? I mean, since we know the central dogma that DNA makes RNA, which makes proteins, proteins are what you actually, you see, right? So like, for example, in sickle cell, there's one amino acid that's changed and you see something completely different, right? You either have a round donut shaped red blood cell or you have a sickle cell one. So it's amazing that one amino acid can do that. But it was a major challenge. I mean, it really was, because in order to go back to Mendel's work and some of the other people that in the early 20th century that were doing, Morgan, like with the flies, and then after Mendel, you really had to know, like here's the one about Morgan, so first you had Mendel's work, right? And he's over here showing, using pea plants and showing this dominance and recessive, and he had no idea what chromosomes were. He called them unit factors. It was either this or that. His terminology was dominance, recessive, independent assortment, and segregation. Okay. Very closely after him came Morgan. So Morgan's group, they worked with Drosophila, and they basically showed that these genes are located on chromosomes. So you had the chromosomal theory, was what it was called. But again, they knew, it's not that they didn't know that there was DNA and they didn't know that there was protein. They just became candidates for the genetic material because we were nowhere near in terms of molecular biology, than where we are today. So molecular biology is relatively new, okay? And so this experimental organisms, you get into Griffith, we're going to go through some of the people. So there's a lot of people in terms of discovery and of DNA being the hereditary material, okay? And also, you know, viruses, we know that viruses are actually not living. Okay, so today, viruses are not living. I mean, we know that they're not living. Back when I was an undergraduate, for us, we learned that viruses were living. So, you know, again, these are theories. These are, you know, what was valid or what we thought, you know, 25, 30 years ago may not be what you're learning necessarily today. And that's something that I find fascinating about science personally. So here we have a little bit, we kind of go through, and I love to talk about some of the people that are involved. And so we have the DNA, so it's like the search of DNA, you know, begins with Griffith. And Griffith is around in 1928, and he works with these two bacterias, and one of them basically, they were pneumonia, and one of them is going to kill the mouse. Okay, pathogenic means kill or make sick. And then there was another strain that wouldn't, you know, wouldn't harm the mouse. Okay, and so his experiment is just pretty much summed up right here. I like this slide a lot. Okay, so this is Griffith's work. So you've got the living S's, right? We know that those are pathogenic, mouse dies, right? You inject it with mouse dies. You have the control that's the R, harmless, healthy mouse, right? If you put heat on these, okay, makes sense. So you kill them, okay, then great, mouse lives. But then when you put what's called a racemic mixture together, so he put a bunch of the heat killed ones, okay, which caused the mouse to be healthy, plus the good kind, right, mouse healthy. When he put them together, the mouse died. Well, what in the world, right? So what is going on? Okay, so that was kind of the first one, and he called this transformation. That's where the term transformation comes from. And this is basically where you have a change in genotype. Okay, it's taken in, so some cells take in, right? They take in the other cells and they create something different. And because of that foreign DNA, it changes the phenotype. In this case, the phenotype is either life or death. Okay, right? So this is a phenotype. Mouse dies, that's a phenotype. Mouse healthy, that's a phenotype. Okay, so keep in mind that phenotype doesn't always mean, you know, brown or yellow or purple and white. It can mean life and death. All right, so moving along, in 1944, you have the Avery, McLeod, and McCarty experiment. Okay, and they're going to actually, so they're going to use Griffith's work. Okay, but this is when they would actually announce that the transforming material was DNA. Okay, so Griffith did the work with the pneumoniae, with the mice, but it would be Avery, McLeod, and McCarty that would actually come along and show that the transforming bacteria was actually, you know, it transformed and it was bacteria, and that it was DNA. But even though, you know, think about, this is 1928, this is 1944, many scientists still remain skeptical about DNA because they don't know, it's like it exists and they know what it is, but they don't know what it looks like. Okay, they just, there's like pieces and parts out there. So you kind of keep going, all right, and more evidence comes up. You get into the Hershey and Chase experiments, and they use bacteriophages, okay, and they actually show that it was DNA and not protein that was the material that transformed, okay. And then here's a picture. They look like little spaceships, okay. And so bacteriophages, they are viruses. So these are viruses that actually attack bacteria. So again, everything in nature has its own enemies, okay? So in 1952, here we have Hershey and Chase, I was just talking about, and they use these phages and they show, okay, so they basically use these bacteriophages and attack E. coli, and they showed that it was the DNA that was the genetic material. And here's a picture. Kind of looks gross, but this is a picture of what they look like. And so you ask, well, how did they see that? Well, they basically back then, things were radioactively labeled, and so they knew, right? We know that DNA has a phosphate backbone. Okay, so P32 was a radioactive phosphorus that would bind to the phosphate backbone of DNA. And in protein, we know that there's those sulfide bridges and that a component of it is sulfur. Okay, so they radioactively labeled it. And these are two very different, I mean, this is pretty different. And they're showing you this is blue and pink. They're not really blue and pink. But they're showing you that indeed it was, the DNA that was the source. Okay. So, I mean, it just keeps going from there. At the exact same time, you've got Chargaff. Chargaff is the one, we have Chargaff's rules, and that gets into the A and the T that go together, and the C and the G that go together. So I'll let you read that, but most of this, so there's Chargaff's rules. And so by the time 1950 rolls around, okay, so we know, DNA's got, it has a nucleotide, it's got the nitrogenous base, the sugar, the phosphate group, that's what makes up a nucleotide. And it's starting to become, you have all these people, you have all this evidence, you have 1944, 1947. So really, in my personal opinion, I feel like Watson and Crick had quite a lot of help, although they would be given almost complete credit for the model of DNA, which, I don't know if I agree with all that, but whatever. So one, and we're talking about another person. I love to bring up some of the people that I call like the underdogs who weren't necessarily given a lot of credit. Are Franklin and Wilkins, and they worked together and they used the technique. So chemistry and biology we talked a lot about. You're probably wondering, well, I don't know why the second chapter has all this chemistry in it. Well, crystallography is actually used in chemistry. It's more of a chemical technique, and it's used to study the molecular structure, right? And so Wilkins and Franklin's would use this, and they would come up with this picture here. This is a picture of Rosalind Franklin. She's one of the first. If you've noticed, I've been talking a lot about males, 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 males. And then finally we have a female that comes along. Let's hear it for the girls. And this is what the X-ray diffraction, this crystallography would look like. Well, it looks like an X, doesn't it? So if you want to know why we depict chromosomes in an X-like fashion, this is the reason. I think that's really cool. And this just talks about it. And it was these images, even though, again, they weren't given a lot of credit, that enabled Watson to deduce that the width of the helix, and this would help give information about the spacing. Because when they first built the model, Watson and Crick, they didn't build it. They built it with like A binding with A and so on, T with T, et cetera, et cetera. But when they couldn't, when they did that, okay, they already had Franklin and Wilkins's work, and they were like, wait a minute, something's not right because the spacing wasn't right. Okay, the spacing wasn't right. None of this is arbitrary. Okay, so instead, it was the pairing of the purine and the pyrimidine that resulted in the consistency with the X-ray crystallography. So a purine and a purine was too wide, a pyrimidine and a pyrimidine was too narrow, and now you have the purine and the pyrimidine, and you have, that was what the X-ray would look like. So anyway, so they made, they determined, not to take away about what Watson and Crick did, they actually kind of put everything together. That's really, in a nutshell, what Watson and Crick did. They put everything together. And for that, really that's their credit. It's not so much that they single-handedly figured out that DNA, that this was the model and so on and so on. So anyway, so this is just some, you've seen these before. This is what it looks like. You've got the double helix here. You've got the anti-parallel, so 5 prime, 3 prime, 5 prime, 3 prime. So you read, so this strand you would read TGCA, and this one you would read TGCA. Okay, because again, they're anti-parallel. And you have to keep these features, have to keep this in mind. Otherwise, when you go to read DNA, you will be confused. All right, you will be confused, okay, when you try to read it. And if we could get through the class without you reading it, that would be fine, but that's not exactly how biology works. And there's lots of different proteins involved in this. I mean, we're trying to make it as simple as possible, but as you know, it is anything but a simplistic process. So these are called complementary, right? They complement each other, and each one of them can act as a template for building a new strand in replication, okay? Each one of them, okay? So you have replication. So this is just the first part that we get into is how DNA replicates, okay? That's all we're talking about, okay? We're not talking about DNA to make RNA to make proteins at this point. We're just talking about how DNA makes more of itself. All right, so first things first, you got to have a template, right? So you've got your double strand, and they partner up A and T, C and G, and they unzip, okay? Unzip, right, like this, zip, zip, doop, doop, doop. Okay? So that's the first part that has to happen. And then once you have the first part, then each parent, so this is a parent strand, or old strand, this is new strand, or baby strand, however you want to term it, then each one of them can make another double helix, right? And when it's all said and done, you end up with, so you started out with one, unzips itself, and makes 2 new ones. And so this is a very nice representation, or I think so, based on old and new, old and new, okay? And this is called the semi-conservative model of replication, okay? Now, you get into here and it shows you, this is the Michelson and Stahl experiments, okay? So we've got a lot of people when it comes to DNA and genetics and all of this, okay? So if you keep the people straight, you will keep the experiment straight and it will help you, okay? Just don't try to memorize everything willy-nilly. Keep in mind what the people did, all right? So this is the Michelson and Stahl experiment. So again, they're asking themselves, with everything we do, there's a hypothesis, right? So the question becomes, well, does DNA replicate? Is it conservative model? Is it conservative? Is it semi-conservative or dispersive? Okay. Now, we know that it's semi-conservative, but how did we get there? Well, we got there because Michelson and Stahl, they made the hypothesis that it was a semi-conservative model. Fine. So how did they support it? Okay, they didn't prove it necessarily, but today there's a lot of evidence that supports it. Believe it or not, this is not a proof. So they labeled the old strands, or the parent strands, with this really heavy isotope of nitrogen. Fine. And then they labeled anything that would come off that was new, and it would have a lighter isotope. Okay, so this is something that makes a great centrifugation experiment. Why? Because if it's heavy, it goes to the bottom. If it's light, it goes to the top. Okay, and a picture's worth 1,000 words. So you've got your heavy, you've got your light, right? You're going to centrifuge it. And with the first replication, right? So these are just rounds of replication. So the first replication, conservative model was out right away. And you can see that the line was in the middle. Okay, so this is all the DNA, right, is right here. Well, so that kind of said, well, it could be semi-conservative or it could be dispersive. All right, well, we better do a second one. So they did a second one. Well, now you start seeing the separation. So this one, the conservative one was already out, but they just showed that it was even farther out. The semi-conservative model was still a go. And the dispersive model, if it was dispersive, it would have gone up to the top. And there were two in this case, not one. So that one was out. And then you have the evidence that supports the hypothesis of the semi-conservative model. Okay. So we get into it in terms of a closer look. Okay, so that was just all background. That's how we got there. Isn't that crazy? A lot of people, a lot of information. Amazing what was going on between 1900 and 1954. And there's just so much stuff. It's amazing that any of the DNA, honestly, can replicate itself accurately and with the amount of speed that it does. It just kind of blows my mind. And I appreciate how well and how accurate it is. So now we're going to get into, so right here in slide 34, we get into a lot of detail, okay? And I don't ask you all of this stuff by any means, but it doesn't make sense to kind of give you a 6th grade level, if you will, or an elementary view of the replication of DNA, because there's just nothing that's like easy about it. You know, I would have to leave out 1,000 things if we gave you a really high level of replication. Okay, so when it starts, okay, you have, it's going to open up, and so here you have, this is 3 replication bubbles are visible along the DNA. No matter what kind of DNA it is, but this is what it looks like. Okay, so you get this origin of replication, and you have a bubble. Okay, and it proceeds in both directions. And why would it proceed in both directions? Because you have 5' and 3' on one side, and you have 5' and 3' on the other. Okay, and you have the old strand or the parental template strand. You'll see lots of different words for this: template strand, parent strand. And then the new one is labeled like daughter strand, just like a daughter cell. See, so some of these names start being the same. And then these are called, like, where that's going is called the replication fork. And then when it's all said and done, right, so you've got the old and you've got the new. And there's a bunch of enzymes and stuff like that involved in this that we'll get into. But for now, and in eukaryotes, the replication, there's lots of different sites like this. It's not just one, okay? Because the DNA is linear and it would take all day if there wasn't multiple sites. So it's, but in a prokaryote, right, it's round. And so there's usually just one site of origin of replication, and it just goes around in a circle. So, so moving on to the DNA polymerase. So you have to say, well, how am I going to add these nucleotides on, right? How am I going to get these letters A, T, C, and G added on? And you have these things called DNA polymerases, which makes sense. ACE means enzyme, polymer means long, right? And it's DNA, so it's doing something with DNA. And this is going to make it longer. So each one of them is added to the growing DNA strand, okay? And it just keeps going. It's very fast. This is just amazing, okay? Here's a picture. I love the pictures. So you have your 2 strands right here. You have your old strand 1 and old strand 2. And you have or it's just taken 1. So template strand. So you have 5' to 3' end. And it always adds these to the 3' end. Okay, so it's growing. Remember, they're anti-parallel, so it's adding them to the 3' end. Okay, so when you read the new one, you're doing polymerase, you have ATP, favorite ATP. Okay, when you read this one, the new one, you read it this way. So it's A, C, G, T. Okay, you're reading it left to right, 5 to 3. When I read this one, it's C, A, C, G, T. Okay, that's important. That's very important. All right, so here's some words. I'm not going to read it all to you. But you will want to read this because you'll want to know what these things are called. So in here, you have it, you see, it's going to unzip. You've got a leading and a lagging strand. Okay, the leading is going 5' to 3', right? So here's the template strand, right? It's anti-parallel. It just keeps going because it's 5', 3', 3'. It's adding 3', 3', adding, it's just continuous, okay? On the lagging strand, it's like it's adding it backwards, right? You can see it's adding it here, and so you get these fragments, and these are called Okazaki fragments, and you have DNA polymerase 3, okay, and it adds them, and then you have DNA ligase. So ligase, it ligates things together. It's kind of like tape or glue. And it's going to come in and it's going to stick those Okazaki fragments together, those Okazaki fragments. Okay. All right. And then, and it's moving. You can see where it's moving. So this one's going, that's why it's showing you this line. It's going like that. Okay. Or one, and you're going to have two, an old one and a new one here. And then for this new one, this is the overall direction of replication. But the nucleotides are always added to that 3' side. All right, moving on. Let's see here. So when you talk about DNA synthesis, okay, so DNA polymerase, like I said, it can only add them to the nucleotides to the 3' end. In the beginning, that initial nucleotide strand is a very short one, and it's either going to be an RNA or DNA primer. And the primase starts, it depends on what you're making, okay? If you're making RNA or you're making DNA. These are some pictures of it, and I'm going to let you go through this. You can ask me questions on it. There's lots of animations of this stuff out there. So, and there's other, let's see, some of the important ones here. Helicase is important because it actually untwists the DNA, or unzips it, as I like to say. It's like unzips it, and then the topomerase, which I'm sure I'm butchering the word, but it actually helps not over unwind it, right? So, it's curled and you're straightening it out, but you don't want to do it too much because it'll break. And you have primase, okay, synthesize actually makes an RNA primer at the 5' end of the leading strand and the Okazaki fragments. There's a bunch of them. Here's DNA polymerase 3, DNA polymerase 1, DNA ligase. So these are what they do, and they're really just definitions. And then to go along with the definitions, they're pictures. Okay, so we already talked about the bubble. Okay, so you've got an origin of replication, you've got a bubble that happens, you've got the leading strand up here, and then the lagging, right? And so the leading strand, so this is basically, if I take this picture and I blow it up, if the leading strand, which is continuous, it's going to go towards the replication fork, right? You have DNA polymerase 3. Not much is happening in the leading strand. It's nice and easy and clean. Then you got the lagging strand, and as the name indicates, it's kind of going backwards. And so you've got these Okazaki fragments, and you've got the primer that has to come along, and it's sticking them together. And you've got your DNA polymerase 1 that's adding the nucleotides and helping to stick them together. And it actually removes the polymerase 1 actually takes off that primer that it put in there and replaces it on the 3' end so that the DNA polymerase 3 can actually add the nucleotides. So this is a little bit of a process here. And then you need something to actually hook it together. So you got the ligase. So the lagging strand a little bit of a process right here. But in terms of everything I was talking about earlier that's on this slide, this is a good picture of it. So hopefully that'll help. All right, let's see. So we already know that it's not stationary at all. Hopefully you don't think that. It certainly is nothing but nothing. It would be not a good idea if we didn't talk about proofreading and repairing the DNA, because as you know, an error in the DNA can cause an error in RNA, which can cause an error in the protein. Also, just having a DNA or an RNA error moving into a protein can cause. So there's so many places in this process, not just in DNA replication, but in the whole process that can go wrong and cause problems. So here's an example of a repair, right? I mean, DNA, it can break. You know, these things are not perfect by any means. And so what you hope is that it's caught and repaired, right? And doesn't move on to RNA or a protein and cause a bigger problem. But we know that that's not always the case. So we have, let's see, I don't go into a lot about the ends. See what else we want to. Telomere, you know what those are from looking at chromosomes. Those are the ends, okay? And they are very, very important, okay? So I want to give them their fair due. They don't prevent the shortening of DNA, but they do postpone the unraveling of the ends of those chromosomes. And something interesting that has been researched is in the process of aging, it is known that the longer chromosomes are, the less aging of the organism there is. So this is a big area of study because of aging. Big money maker, you know, how can I get rid of my wrinkles? You know, all those things, my gray hair, you know, all that stuff, right? You know, I want to have my youthful glow. So as we get to the end, so those are the telomeres. You can see them there. They've been highlighted. So there is an enzyme called telomerase that actually helps lengthen those telomeres in germ cells. So this is a big area of study. All right, so this takes care of chapter 16. Let's see if I can make it small over here. Okay, well, let me go to 17. Oh my. Sorry, let me move this around so I can close it down there. All right, what are we at time-wise? We're at an hour and a half. Well, I'm going through 3 lectures, so this is probably why you're lucky that you're not in front of me all the time in class. Okay. Let's see if I can get the animation up here. All right, gene to protein. Okay, so we've gone over a lot. I don't know about you, but I'm tired. So if you want, you can always press pause and come back to this video. I know it's going to be very long, but I figured it was better to kind of get through it. And if you want to, you can kind of go through it in its entirety, or you can break it up into its different chapters, however you want to look at it. Okay, so the last chapter is 17. So this is where we go from gene to protein. Well, what does that mean? So we've gone through genetics, or at least a little bit. We've gone through DNA, how we got there, who discovered what, the people involved, why do we know what it looks like. We've gone through replication in chapter 16. So now the question is, well, how do we get from actual DNA or gene to the protein level? Okay. All right. So, you know, as an overview, we're going to bring in some new words. Okay. So we have transcription. So DNA making RNA, right? And then we have translation. So these words, transcription and translation, very, 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 very important. Okay. Translation is how you make a protein from the RNA. Okay? And here we have a very complex protein. All right. So we get into, again, we're going to bring in more people and we're going back in time again. Okay. So we're going to say, well, in terms of the central dogma, and the central dogma is another example of, you talk about something that has really evolved or morphed over time, and it is a central dogma. When I was in school, back in my day, it was thought, and it wasn't that long ago, so it was thought that DNA strictly made RNA and RNA strictly made proteins. Wow. Well, we know that that is not the case. So we're going to get into that a little bit. But at the same time that Mendel was over there working on peas, okay, in Austria, we have a British physician, so Dr. Garrod, and he would be the first one to suggest that a gene, now again, we don't, at that time in 1909, they didn't know what a gene was made out of, right? But all this work was coming out about genes and alleles and unit factors, and we had Morgan doing his work and saying that genes were located on chromosomes. So he comes along and he says, well, maybe phenotypes, okay, so that word is already out there, are actually coming about by an enzyme. So now we're getting into enzymes, okay, because protein chemists, okay, proteins, right? So what did they think the first DNA material was? The genetic material. They didn't think it was DNA. They thought it was proteins. Well, enzymes are proteins, right? So they thought, well, it makes sense. This kind of makes sense. So he says, well, it must be that there's a certain amount of enzymes that go together and catalyze these chemical reactions, and that somehow is dictating some of these inherited diseases. Okay, so that's what he was interested in. He was a doctor, so he's looking at it from more of a disease perspective. Okay, so then meanwhile, you have Beadle and Tatum. So Beadle and Tatum, they're working with mold. Okay, because everybody, again, not every biologist or protein chemist or chemist works on the same thing. So, like, for example, when I was in graduate school, I worked on maize, corn. A doctor who's looking at diseases is not going to be interested or studying flower phenotypes, but he is interested in the phenotype of life and death or a recessive disease. So Beadle and Tatum, they worked with Neurospora, okay, which is just a type of bread mold. And what they did was they created these mutants that would show, by crossing them and seeing what mutants lacked what enzymes, then they actually developed the one gene, one enzyme hypothesis, okay, which basically stated that one gene dictated a specific enzyme. So you might be scratching your head, okay, I can see you sitting there like, oh, what? All that's saying is that one piece of DNA, so if I've got one piece of DNA, AGCT, let's say, okay, that AGCT is the directions for one protein, okay, because we know that enzymes are proteins, right? Okay. So, and this is just a picture of what they did. So there was some media, and they made some mutants, class 1, class 2, class 3 mutants, and they made them with different amino acids. That's all this is. And the ability, again, life and death, right? So if I mutate them, if I mutate class 1s, then, and I don't have the right precursor to make the enzymes, then it won't live. Okay, that's how this works. So gene A, enzyme A works, okay? Now we have it. So these are wild type. Wild type means found in nature. Does it mean dominant or recessive? No. It means mostly found in nature. That's what wild type means. Okay? It can be dominant, but it doesn't necessarily have to be. Now we take the class 1 mutants. Okay, so there was a mutation in gene A. Well, mutation in gene A doesn't live, right? B doesn't live. C doesn't live. So this is the one gene, one enzyme hypothesis. Okay. So now we keep going. All right. So all enzymes are proteins, but not all proteins are enzymes. That's a mouthful. And you know that because of chapter 4, when you study macromolecules, you saw how many different things are proteins. So they later revised the hypothesis and made it one gene, one protein versus one gene, one enzyme. Okay, because we know that proteins have several polypeptides in them, and each one of those has its own DNA sequence. Okay, so moving on to transcription and translation. So transcription, you've got your blueprint from your DNA and you're going to make your RNA. Translation, you've got your RNA and you're going to make your protein at the ribosomes. Okay, we know that too. All right. In a prokaryote, this stuff happens very simultaneously because what is missing in a prokaryote? A nucleus. Okay. In a eukaryote, right, you've got the separation, so you've got transcription happening in the nucleus and translation happening outside of it. Okay, so a little bit more complicated, but that just has to do with the fact that eukaryotes are a little more complicated. So here we have prokaryote, boom, bada bing, bada boom, right? Boom. Everything's happening all kind of doop, boop, 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 boop. And then you've got the eukaryotic cell. Okay, so nice and easy over here. And now you have the eukaryote, so you've got your DNA, okay, got your transcription, it's going to unzip, you're going to get your pre-messenger RNA, you're going to have some processing, you're going to get a head, a tail, messenger RNA, and then it's going to go outside of the nucleus, it's going to find a ribosome, and it's going to make a polypeptide chain, right? Okay, so the genetic code, right? Well, there are 20 different amino acids, but there are only 4 nucleotide bases, right? A, G, C, and T. So goodness gracious, how do we figure out what it does? Well, that's when we get into the triplet code. So for those of you that like codes or puzzles, this is where we come to. Okay, so they basically, the smallest units of them, just 3, so 3 letters, makes up an amino acid. Okay, that's all this is saying. Okay, and so you read them. This is what I want to point out, is that whenever you're reading DNA, you read in 5 prime, 5 prime, start with 5 prime, go to 3 prime. That's how you read it, like read the letters. In translation, is going to be the same thing. You're going to read in 5 prime to 3 prime. And the reason that I'm harping on that is because that is where everybody gets it wrong. Okay, they end up reading it backwards. So if you're having trouble with this, turn it around. All right, so I love this slide. This is probably one of my favorite slides in the entire chapter. Okay, here's your DNA. All right, gene 1, gene 2, gene 3, gene 4, whatever. All right, you take the DNA and it unzips. Okay, now when you are making DNA, RNA, and protein, okay, it uses a template strand. All right, each gene uses a different template strand. So we're not replicating. Okay, there's not 2 template strands and they're coming apart and then both of them are It's not doing that. It uses one side. Okay, so gene 1 might use this side and gene 2 might use the other side and gene 3 and so on and so on. So in this one we've got a template strand. You read this way. So T, G, A, G, C, etc. Okay, 5 prime, 3 prime. You have transcription. It's complementary, right? So don't forget. So when you read the messenger RNA, it's UGG, UU, because there are no T's in RNA. So what pairs with A? Uracil does. So it's a U. What? Now C and G, they're the same. U pairs with A. Okay, do not forget that. That's a rule. There are no T's in mRNA. None. Okay, so how do we read this? We read this way, 5 prime. It finds so it gets processed. This is all happening in the nucleus. It gets a hat and a tail and splicing and all kinds of stuff happens to the mRNA so that it can go out. Now you may be wondering, well, why does it have to go through this complicated process, the messenger RNA? Because it's going to go out of the nucleus. It's going to be attacked. There are things in a cell called RNases, which destroy RNA, okay? So this thing's got to be protected in case something tries to attack it. Otherwise, what's the point? Okay, what's the point of creating this great, DNA and RNA and having this great messenger if the messenger is going to get killed when he goes outside of the house, right? The nucleus. So while he's trying to find the ribosome. So as you can see, this is translation. And so these three, so UGG corresponds, this is a codon, to the trip amino acid. And you also know that at the site of the ribosome, there's an anticodon. So the only thing that's not on this sheet is an anticodon. Well, the anticodon is going to be the difference. So it's going to be an A, C, C. Anticodon means against, right? But what corresponds to the actual amino acids is the mRNA. So you're sitting there saying, well, how do I know what the amino acid is going to be? Read the messenger, right? The message is in the messenger RNA. Okay, so here we go again. All 64 codons, so there's 64 codons, like different puzzle pieces, if you will. They were all deciphered by the mid-1960s, okay? But it's redundant, okay? The genetic code is redundant, okay? So no codon specifies more than one amino acid, but you can have a codon, you can have lots of codons that specify one amino acid, okay? And I'll show you what I mean by that, okay? So this is the first base, right? So this would be, right, a codon is 3. So this is the first letter, the second letter, and the third letter. So this is how you read this chart. So if the first letter is C, I look in the C, right? If the second letter is a U, I look this way, and then I look for the third letter. If the third letter is a U, then I have CUU, which corresponds to leucine. 3 other codons, okay, one codon, another codon, another codon, another codon, correspond to leucine, okay? And that's what it's talking about, okay? Now, a start, if it's at the very beginning, then it's going to have an AUG, which is methionine or start, okay? If it's at the beginning, you just say start. If it's in the middle and you have AUG, it's just a methionine, okay, or an MET. And then you'll notice something interesting, that there's 3 different ways to get a stop codon. And stop means stop. So you have to start. There's only one way to start, but there's lots of ways to stop. So you can see that if it's supposed to be, let's say you had a messenger RNA that was supposed to be UAC, but there was a mistake or a mutation, right? Oops. And it was UAA. What would happen? It'd stop. You'd have a premature stop codon, and the translation would just stop. So what would happen with your protein? Well, it would look funny, right? So you can see that I find this fascinating because you can see how just one change, depending on where it's at, especially with the stops, it's at the end. But there's this thing called wobble, okay? We can look at it with leucine. We'll just go back to our leucine. Okay, notice that 1 and 2, first letter, second letter, are all CU. CU, 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 CU. The last letter is UCAG, so all 4 of them. Well, if it makes a mistake there, right, let's say it was supposed to be a U, but it ended up being an A. Well, that's okay, because you still get leucine, right? It's the amino acid that matters. But that's a different situation if you're dealing with something else and you've got a UA, and you get an A or G instead of a U and a C. Well, now it gets kind of dicey, right? So it just depends on what you have. I mean, it really depends. And you know the genetic code is universal, and you can, that's why you can transcribe and translate from a like a fly, fly or pig, and vice versa, right? So here is, you got a tobacco mosaic plant, and if I put like a lightning bug gene that lights up into it, right? What's it going to do? It's going to light up the plant. Okay, isn't that cool? That's where you get like genetic engineering and stuff like that in it. All right, so this is going to get into details on transcription. Okay, so a lot of this, it makes sense, right? So if you're making RNA, are you going to use a DNA polymerase? No, you're going to use an RNA polymerase, right? And the same rules exist except for that U and T. There is no U in RNA. Okay, none. All right, and the process starts, the DNA sequence that you have, so you have your template, and then the RNA polymerase comes over to attach to it, and that's called a promoter. Promoter means one. And then it just starts transcribing, making, just like you kind of did in lab, like with the beads, right? You can look at it with beads. You can also write this down on paper. There's more than one way to do this. And so here you have the promoter, right? So here's your DNA. That should be real familiar for you. You've got the transcription unit, okay? You've got promoter, that's where it's going to start. The RNA polymerase comes over, sits on it, right? There's start. And then you have initiation. You've got that unwound DNA, okay? Probably have some helicase in there. And you've got your RNA, so they made it a little bit of a different color, which is nice. You've got your RNA polymerase, and it's adding in. And then you've got your elongation, okay? So this is all it's showing you. That was the first one. You have initiation, and then you have elongation. And the elongation is spitting things out. And what it's trying to show you, albeit sort of not as great in this picture, is that, again, things are added to the 3' end. So you started out, and you can see by looking at it that the template strand, so keep in mind that this bottom one is the template strand, okay? So it's 5', 3', but when the completed RNA transcript is reversed. So this has to be the template strand. And then your polymerase, your RNA polymerase is done, so it just falls off or comes off and gets ready to do it again. Now this guy here, the RNA transcript, is ready to be processed, if you will. So here's a little bit of a blow up of it. Okay, there's the template. It has to unwind. That's where it kind of gets confusing. So it unwinds. And then you can see the 3' end. It's adding the RNA nucleotides. This big old thing is the RNA polymerase. Okay. All right. And so 3 steps. You have the beginning, initiation, elongation, and the ending. Okay. I think a couple of the quiz questions, or at least one of them, maybe even a test question, has something that talks about a Tata box. And the reason that that's important is because in eukaryotes particularly, the promoter is actually called a Tata box. And the reason why it's important is because it signals, like when it sees a Tata box, okay, it says, hey, start right here. That's why it's important, okay? And you can read some of this other stuff, but I like the pictures. You can tell I keep going to the pictures. So here we have that same, it's no different than what we just saw, but you have the promoter region. Okay, so same picture from before, but you can see now that we put a Tata, so T-A-T-A-A, T-A-T-A-T-A, Tata box. Okay, and then you have some transcription factors, some bubbles, and there's lots of things. This is not an easy process, but it's just trying to show you. And then you have that exact same picture. So you have your RNA polymerase right there. That's this big old guy. You got some transcription factors. These are helpers, and they help, kind of get things started and move along. All right. So when it ends, in prokaryotes, basically the polymerase just stops transcription at the end of it. I mean, there's really not a lot. But in eukaryotes, it's the pre-mRNA is cleaved from the actual RNA chain, and so it just eventually falls off. That's all that really happens. And then after transcription is over, there's some splicing, and the messenger RNA gets a head, a cap, and a tail, a poly A tail. Okay, so this is it. So 5' end, so we've got a hat and a tail. And I like to think about that. People ask, well, why would it do that? Well, when you go outside, right, and it's cold outside, you put on your hat and you put on your boots, right? I like to think as the messenger RNA to get modified so that it doesn't get eaten up when it goes outside, okay? And so here's a couple of what it would look like. So here's your cap. It's just some phosphate at the top. You have your untranslated region, that 5' UTR. That just means that there's nothing really going to be translated there. There's no information here. Then you have your start codon. If you remember, that's your AUG. And then you have your stop codon, and that's one of your other three. Then you have a whole bunch of poly A signals, and this is a 3' UTR region, so this is not translated either. And then you've got a big poly A tail. So the system is designed to really protect the protein coding region right here, right? So it has lots of things in the front of it, the 5' end, and lots of things at the 3' end to make sure that nothing happens to it when it goes out of the nucleus for translation. Okay, so the splicing, and you have introns and exons. So exons, those are your coding region. Okay, your exons. So exon those are the ones that are going to eventually be expressed. Okay, the introns, they get spliced out. Okay, they get removed, and then the exons get joined together. Okay, so those are two words that you need to know their meaning. So here, let's go back to our picture. You have your pre-messenger RNA, like we just talked about. You have your cap, you have your tail, right? And it's kind of blowing up that middle piece. And you have an exon and an intron. Well, what it's going to do is it's going to cut that intron out and put those exons back together again, because it doesn't need this. So anything that doesn't need to be there, that's not going to be translated, it's gotten rid of. Okay. And there's various ways to do this. It can be splicing with spliceosomes, and I like to think of them as they like splice or slice. There's snippers, that, as I call these snippers, that these are small nucleotide ribonucleoproteins, kind of a mouthful, that recognize the splice sites. So here's just one example of it. They kind of come in and it loops. DNA loves RNA and DNA. They love to fold or make a loop, right? So the system takes advantage of that, and kind of, without stretching it out and cutting it like lengthwise, it makes a loop and then close together, kind of splice, and then the intron is out. So these are just visual pictures. Ribozymes, introns. So there's some information here. So this is another picture. You have your DNA, your gene, right? You have your transcription, great. We're gonna make RNA. You have your processing. It gets the hat, gets the tail. Introns are out, et cetera, et cetera. You have your translation, so it finds the ribosome, and now you end up with your polypeptide, which are your different domains. So you can tell where they're coming from. So this is coming from exon 1, exon 2, and exon 3. So that's a nice visualization there. All right, so let's get into translation. Let's see here, we've got a picture. So here we have your messenger RNA, okay? So in terms of its movement, it's moving this way, all right? And it's going to attach the anticodon, all right? That's what that, so there's a transfer RNA, and attached to the transfer RNA is going to be the amino acid on one end and the anticodon on the other. And they just come in, they just come in and they hook on, and then they read it, and they're reading it, okay? When they read it, they're reading this messenger RNA 5' to 3'. Okay, so it's UGG is one codon, UUU is another codon, and GGC is another codon. All right, and it's in that order. Okay. All right. This is a little bit more probably of an actual looking picture than our animated version, but I like our little candle version. Okay. So this is, really the symbol, different books use different things, but this is really what it looks like. You've got your anticodon, okay, and you've got your amino acid attachment site, and this is your tRNA. Okay, so there's just two steps in translation. Okay, that's all it requires. You better match the right tRNA and amino acids. So, if you've got glycine, you better match the two that belong together. Okay, otherwise, well, that could be a problem. Okay, and you have to have the correct match between the anticodon and the codon. So, if you have AAA in your codon, then you need to have what in your anticodon? Well, you need to have UUU in your anticodon. And remember, what does it read? The system always reads the mRNA. Okay, please remember that. All right. So here's another picture. I'm not going to go into all this. We'll be here all night. Ribosomes, basically what I want you to know about them is that they're responsible for protein synthesis. And, at this point you have to know a little bit more detail about them, but and these, we're not going to go into a lot of the large and small talk. This is real molecular, but I want to show you a little bit about what it looks like, a computer model, because when we draw them, we kind of draw this poofy brown thing, and that's really not what they look like. All right, so these are the 3 binding sites that a ribosome has, and here's kind of what it looks like empty. Okay. And then here's what it looks like. I like this picture again. This is a good one because it's kind of blown up. So it's going to show you. So how do you read the messenger RNA? You're going to read it from this way. Whatever this is, that's 1 codon. 2, 3, 4, 5, 6, 7, etc. Okay, so it's building it. You can see that it's building it as it goes. And there's the growing one. See, there's the next one that gets added. So it's doot, doot, and it just keeps on going. All right, and same thing. Translation, you've got start, elongation, and termination. And what ends it? Well, stop codon, right? Starts with an AUG, and then it's gonna, and this is a nice picture too because it's blown up. So it finds an AUG, or the first AUG it comes to, okay, and that's gonna start it, and it sees it, and it's like, okay, great, MET, and then it keeps going, do, 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 right? Keeps going. Okay. And here's a good, picture of it. Just keeps on going until it reaches a stop, right? When it finds a stop, it's done. Okay. All right. And here's some other pictures just with what they look like. And there's some post-translational stuff on that slide back there. The ribosomes, are identical, and they can, so the ribosomes that are on the ER and the ones that are floating around, they can come on and off as much as they need to, right? It depends on the type of cell. If you have a bunch of free-floating ribosomes, chances are the cell is maybe making something that's going to be exported out or secreted, and so you're going to need a lot of free-floating ribosomes because it's the function of the cell. But if you've got a different function for a cell or a tissue or whatnot, then maybe you don't need so many free-floating ones. You might have them bound to the ERs. That's all that this is saying. Here's some more pictures. This is on the ER. May need to come in. Then it can go off again. And the types of RNA, this is a good one because it just kind of gives you what type it is and what it does. As you can see, there are so many of them. All right. And we get into point mutations. So this is a hemoglobin. So you've got wild type hemoglobin DNA, RNA, and the normal. So this would be, and then you've got the sickle cell. So this is just one little tiny T to an A. That's it. That's all it takes. And you go from a normal hemoglobin gene to a mutated gene. Scary. Okay. Types of point mutations, I believe that you've seen this too, like what kind has, what means more. The wobble, again, the third base has some wiggle room or wobble room, but you can get substitutions, insertions, deletions, um, you know, the nonsense, I like to say the nonsense mutations, those are the ones that nearly always lead to a non-functional protein. And you know that because they don't make any sense. They're nonsense. I just think that's um, an interesting word. Missense mutations are much more common. And they might, a missense, miss meaning like not the right one. So a missense mutation would be, you're still going to get, let's say it's supposed to be tryptophan, but it accidentally encodes for leucine. So it encoded for one, but it's not the right one. Okay. And so you get something very different when it encodes for one, but not the right one. Still might be functional. There's a big difference between that and something that you get a stop code on and your protein doesn't even work. So the substitutions, it makes a difference what they are and what's happening to them. And then insertion and deletion, that's exactly what it sounds like. This happens a lot with chromosomes. You'll see in conditions, you'll see, let's say that something is actually missing. So that's a deletion, like a part of a gene is just gone, or there's an insertion where something else got inserted in. And so that produces, those are usually a frame shift mutation. So frame shift means how you read it, right? So if I'm reading A, and suddenly there's something that gets inserted, then I'm not reading the same information. You have to think of it like a book. When you read, if there's another word in there, well, then the sentence doesn't make sense, right? That's a missense. And if it stops altogether and there's nothing left on the page, well, that doesn't make any sense. So that's a nonsense one. That means that's what we were just talking about before. We get into mutagens. I'll let you read this and some more pictures. Okay. So I think I have gone through 3 different chapters and been on here for almost 2 hours. But these are some of the more important chapters, and hopefully, I hope that this will help you as you go back and you're looking at week 8, and then as you look at week 9, because it kind of goes together. They're very much related to each other, but if you really look at the timeline, I think for me that's when it started to make sense. Is that you have to realize that it's a very much a history of how things were kind of happening either simultaneously at the same time or later on down the line. So we talk about it the way that we talk about it because it really is a historical document that runs from the early 1900s until at the point where we're at. Well, by the time this ends, we're at like in the 60s, 70s time frame. So the technology is not real great, but so we have just done a span in 3 chapters from about 1900 to about 1960. So lots going on. So if you have any questions, I'm going to think about possibly some extra credit, and I have found, let me see if I can find it down here. I did find some practice problems. Maybe this would help you in terms of the genetic problems with week 8 and the quiz that's in week 8. And so I'm going to look for a couple of things that maybe you can do, and you can turn them in for some extra credit, or if you just want to use them to study, that's fine too. But I'll put a couple of points with them. So hopefully this will help you. And if there's any questions on anything, or there's something specific that you're just having a hard time with, then write me and let me know, because it's a little bit easier for me to work with something specific. There's a lot of meat in these 3 chapters. So we'll have to narrow it down a little bit. But I'll put this up in Blackboard, and I'll I'll put some stuff up and kind of tell you, direct you where to go. But hopefully this is helpful, and I'm running out of steam, so I will talk to you all later, and I hope you've had a great weekend. And I did put some information up if you haven't looked at the practice final exam. That's up. That's going to be extra credit. And I think on the 18th, if I remember correctly, the final exam should open up. So hopefully everybody is making their plans. All right. Well, that's all. I will chat at y'all later. Have a good night.